Okay, so since it is already 3 o'clock, we'll get started with the TA session. I'll just start sharing the PPT. Yeah, I hope the PPT is visible now. So we'll get started. So welcome once again to the week 6 of this TA session for the course Bioengineering and Interface with Biology and Medicine. I am Arjun. I am one of the TAs for this course, and yeah, welcome once again to this week 6 session. So yeah, originally this was supposed to be planned to supposed to be held on March 1, that is on Saturday, but due to some unfortunate circumstances, I had to reschedule this to Monday. Um, my apologies for that. So weeks for week 6, this will be the usual routine. So we will go over a general recap of the what happened in the course lectures for this week 6. Following which we will go over the sample problem statements for week 6. We'll go over some sample problems, and then I will be discussing the week 4 assignment questions. So we'll get started now. So what happened in week 6 was a mix of topics compared to last week, where we like focused on genetics, where we ended genetics. So this week focused on a lot of variety of topics. The first topic focused on cell cycle regulation and cancer. So we started the course instructor Dr. Sanjeeva Srivastava started the um, course started this week's lecture with the introduction of cell cycle, explaining how cells have to divide and divide for multiple purposes, for damage repair and for passing on genetic material, etc., etc. And we also saw that there are like two types of cells with respect to cell cycle and cell division, which is somatic cells and gametes, that is the your reproductive cells. And with respect to cell cycle and cell division, there are a lot of differences between how somatic cells divide and how reproductive cells or gametes divide. So uh, basically, to say that there are two types of cell divisions, mitosis and meiosis, that you would have seen in the lectures, and almost all the somatic cells undergo mitosis, whereas the gametes undergo meiosis. And mitosis is like a single step process where you had like four stages in it, whereas meiosis happens in two different processes, meiosis 1 and meiosis 2. uh, One of the meiosis 2 represents almost similar to mitosis, whereas there are a lot of recombination happening in meiosis 1 stage. And yeah, so that's about a general recap of what happens between the somatic cells and reproductive cells with respect to cell cycle. And here you can see a general diagram of the overall overview of how the cell cycle happens in cells. So basically, we have a something called, so basically broadly the cell cycle is divided into two phases. One is the interphase and other one is the M phase. The M phase is where the actual cell division occurs, whereas the interphase is the phase where the cell prepares itself for the cell division. So in interphase we have the G1 phase, G0 phase, S phase, and the G2 phase. So G0 phase is just the dormant quiescent stage where the cell doesn't do anything, whereas in G1 phase the cell grows to accommodate more space for the division of two cells. And in S phase, DNA replication happens where the ploidy of the chromosomes changes into twice to so that when mitosis happens, the division of the chromosomes can happen. And in G2, the cell recruits additional proteins so that it prepares itself for mitosis. And in mitosis also, In the M phase also, the M phase is divided broadly into two phases. One is the mitosis phase and one is the cytokinesis phase. The mitosis phase is where the cellular material divides into two, whereas the cytokinesis is the phase where the cell division, actual cell division happens, where the formation of the actin fibers between the cellular material happens, causing the division of the two cells. And the mitosis phase consists of four small stages or steps: prophase, metaphase, anaphase, and telophase. In other materials, you would also see a fifth stage between prophase and metaphase, which is the prometaphase. So all of these stages correspond just to the migration of chromosomes towards the centromeres, as well as alignment of chromosomes and recruitment of your microtubules and microfilaments towards the chromosomes, pulling them forward, and then the actual division. And yeah, and this is the general um, overview of what happens between mitosis and meiosis. So in mitosis, the ploidy of the daughter cells don't change. So if the ploidy of the parent cell is 2n, the diploid cells emerging from it will also have 2n. Whereas in the case of your meiosis, the ploidy becomes half. So for example, if there are 2n cells, your ploidy of the final daughter cells will be n. Yeah. So and we saw, so this is the general overview of cell cycle, and we saw that in the towards the end of the video we also saw that the cell cycle has some specific checkpoints at each of the G1, G2, and M phase to check that uh, cell division is happening properly. And we also saw that this is happening with the help of specific proteins called cyclins and cyclin dependent kinases, an enzyme called cyclin dependent kinases. So these form a complex, and these act as cell cycle checkpoints. And we also saw that in cases like cancer, where any cell grows uncontrollably, the uh, there is a disruption in this checkpoint, or there is a disruption in the activation or this recruitment of this, either the cyclins or the cyclin dependent kinases. So after we saw about cell cycle and cycle regulation and resulting of cancer, the second video in this week's material was focused on developmental biology, where we saw the initial phases of this development, where the course it took the example of a frog, an adult frog, and how the sperm and egg fuse together to fuse together in a process called as fertilization, and how this fertilized egg undergoes several processes, some of which include cleavage, gastrulation, and organogenesis. So the cleavage of the fertilized egg results in compartmentalization, resulting in the formulation of the blastula, and the blastula undergoes a process called the gastrulation, where the uh, resultant is resultant product is called as a gastrula, which has all the ectoderm, endoderm, and mesoderm, which are called the germinal layers. And here you can detail you can see how the blastula forms the early gastrula, and how the early gastrula undergoes the late gastrula to form the three germinal layers, such as ectoderm, endoderm, and mesoderm. And the gastrula further undergoes something called as an organogenesis, where the organs are starting to form in the gastrula, and resulting it into a formation of a young organism, which is almost the replica of the adult frog. And after that we also saw some processes called as, in developmental biology we also saw some processes called as the apoptosis, which which means that cell is undergoing a programmed cell death. Apoptosis can be beneficial as well as detrimental depending on the use case. And towards the end of the lecture, we also saw several steps which are the basis of which use these concepts we learned in developmental biology to know more about the developing child inside a human. So some of this include a process called as an amniocentesis, where the amniotic sac, where the amniotic fluid from the amniotic sac is taken and is processed to know the gender of the growing baby. And some in very an earlier case of amniocentesis is the chorionic villus sampling, where the samples from the chorionic villi is extracted and it's subjected to karyotyping. We saw what karyotyping is in the last week. So karyotyping is a process which results in knowing the genetic makeup or the chromosomal makeup of an organism. So the chorionic villi can be sampled out of the womb and can be sent for karyotyping. And one more process that we saw, and it's a pretty famous one, is IVF or IVF, where it's a form of external fertilization where the sperm and the egg are collected from humans and they are cultured in an external environment. And once they are fertilized and once they are ready, they are implanted into either into a woman or into a surrogate mother, so that it grows into a baby. And after that, we saw some of the in the third lecture we saw some of concepts from animal cloning, where we initially were introduced to a concept called totipotency, where a totipotent cell is a cell which can be differentiated into any kind of cell. And we also saw, depending on, once again, yeah, sorry. So depending on the potency of the cell, a cell can be classified as totipotent, pluripotent, multipotent, or a normal somatic cell. And we also saw what are stem cells. So all of this stem cell is any cell which can be programmed to differentiate into any cell of our choice. So that can be done using the addition of growth factors and giving it enough nutrients and space. And we also saw how this concept of totipotency helped us to helps us in animal cloning, where this particular scientist, Dr. John B. Gooden, uh, first performed experiments to clone a frog. And we also saw other kind of experiments like the dolly experiment, which focuses on the cloning of animals, wherein the uh, wherein the cells are where the gametes are extracted from a frog and then they are irradiated with UV, and then uh, it is implanted into a and the health and the nucleus alone is extracted, and then it's implanted into the cell of our choice to produce the animal of our choice. And we also saw something about cell reprogramming, as in how normal cells can be reprogrammed to function as uh, pluripotent stem cells, uh, which can be differentiated into any other cell type of our choice. And we also saw some case studies by Yamanaka and Gordon, which focuses on this cell reprogramming. And to unfortunate this thing, we also saw some of the news clippings where people misused this knowledge of animal cloning and stem cell technologies to falsify results and leading them to imprisonment and other grave punishments. And in the final lecture of the video, before the clinician's perspective, we saw some concepts into evolution, where we were first introduced to Darwin and Darwin's theory of evolution. And Darwin's theory of evolution mainly focuses on natural selection and adaptation. So the most famous example that Darwin's theory of evolution cites is the beak size of the birds in Galapagos Islands, where the beaks of the birds are modified to accommodate for their beaks' purpose, whether to cut open fruits or nuts or to just aid them in flying. And after that, we also saw something called as the Hardy-Weinberg principle, where scientists called Hardy and Weinberg, they came up with this principle of evolution that under some certain criteria, which includes no natural selection, random mating, very large population size, no mutation and no migration, there is a formula for arriving at the frequency of homozygous dominant genotype, frequency of heterozygous genotype, and frequency of homozygous recessive genotype, and all of this can be clubbed together to form a meaningful formula. So p square plus 2pq plus q square is equal to 1. And finally, we saw about divergent and convergent evolution, where in divergent evolution there is a common ancestor, and yeah, there is a common ancestor, and uh, the common ancestor gave rise to different types of animals, which obviously is due to adaptability and use of the animal. And in convergent evolution, different animals which are not related to each other are coming together to form a singular organisms or organs with similar function. So that's all about this recap for the week 6. So I'll have a short doubt session like till 3:20 or something. So after that we can start discussing the sample problems for week 6. Okay, so since it's 3:20, we'll get started with the uh, problem statements for this week. So we'll get right into it. So yeah, so most of these questions are like from directly from this either from the course material or from the lectures. So it's kind of easy to answer. So question number one, genomic instability is thought to be a hallmark of cancer and can result from dysregulation of the cell cycle. Apart from genetic mutations, chromosomal misaggregation during mitosis is another driver that leads to genomic instability. What cellular structure is primarily responsible for ensuring accurate chromosome segregation during cell division? So the question is, what cellular structure is primarily responsible for ensuring accurate chromosome segregation during cell division? So out of all these four options, like you can see only like microtubules is like the organelle that is involved in cell division as well. So the option is microtubules. So microtubules are those organelles, cellular structures that hold the chromatids from each end, and then they pull towards them during metaphase and anaphase, which results in the segregation of chromosomes for each daughter cell. So I'll move on to the next question. The two major types of cell division processes take place in higher eukaryotes. One happens in almost every cell, mitosis, and another is a specialized type of cells, that is meiosis. Both occur through a number of successive phases. Please choose from the diagram that best reflects each type of cell division and its phases from the figure below. So I'll read out the question again. Two major types of cell division processes take place in higher eukaryotes. One happens in almost every cell, which is mitosis, and another is a specialized type of cells, meiosis. Both occur through a number of successive phases. Please choose from the diagram that best reflects each type of cell division and its phases from the figure below. So they're asking us to like classify which phase, whether mitosis or meiosis, or which are the phases that is present. So uh, this one is also like pretty like simpler to solve. So you can see here that both the sister, both the chromatids are like intact in one of these this thing. So this should this should result in like meiosis. And so since we have like four, the ploidy of the chromosome is four, so we will have like four different chromosomes here. So but in case of mitosis, the ploidy will still be two. So you will be having only like two chromosomes here. So from that itself, we can see that it is that is we can see that the process is called meiosis. And since the chromosomes are still at the center of the plane of the cell, it can say that it's metaphase. If it's anaphase, they will be pretty far from each other. And since the first option is like meiosis metaphase, so there is like no other option except that which represents meiosis and metaphase. But even in Sega, even for figure number 2 and 3, you can see that 2 represents mitosis anaphase, and here also 2 represents mitosis anaphase, and 3 represents meiosis anaphase, and here also 3 represents meiosis anaphase, which is like a next step of this meiosis metaphase. So you can also see here that next step the chromatids are pulled closer towards each central here. So the correct option is one is meiosis metaphase, 2 is mitosis anaphase, and 3 is meiosis anaphase. So I'll go to the next question. So, in a diploid cell with six chromosomes, how many unique combinations of chromosomes can be produced in the gametes through independent assortment during meiosis? After the first meiotic division, how many, yeah, how many chromosomes will each daughter cell have? So, as we can see, the second part of the question is a bit easy, I guess. So we are having we are having a diploid cell with six chromosomes, and they are saying that it undergoes meiosis, and after the first meiotic division, they are asking how many chromosomes will each daughter have. So automatically, after the first meiotic division, it will become half number of chromosomes in each daughter cell. So the number of chromosomes will be 3. So it will be either this option or this option. And out of this, we have to find out how many combinations of independent assortment can have. So that is 2 power 3. So we have 3 chromosomes after the first meiotic division, and the number of combinations that can be produced through independent assortment is 2 power 3, which is 8. So the answer is 8 unique combinations, 3 chromosomes. So I hope this is clear. So I'll also upload this video to. I'm recording this lecture right now, so I'll also be uploading this to YouTube. So in the case where like you have any doubts, you can also post it in the YouTube comment section. So question number 4. Cyclin CDK complexes are essential cell cycle regulators. Cyclin D and CDK4 combine in the G1 phase to start the S phase development. The question is what does the cyclin D CDK4 complex do primarily during the G1 phase? So this is like directly from the material. So the answer is activation of DNA replication, because S phase has to S phase's job is to replicate the DNA. So the G1 checkpoint preceding the S phase will be to activate the DNA replication after it has found out that no all the nutrients have been recruited to form to do the S phase. So I'll go to the next question. So the diagram shows the events happening during the development of an embryo. Pick the correct options for 1, 2, 3, and 4. So they have asked. So we have a process where the embryo is being developed. So we have to. So we have to find out which of the processes will lead to form. So if we have a sperm, sperm entering an egg, so it forms a zygote. So we have to see what process is it that will fill one. So automatically, if you know that the sperm fusing with the egg, then it's called as a fertilization. So one will be fertilization, and two is cleavage. So because your zygote is undergoing cleavage to form the 8-cell stage, and your yeah, from this itself we can identify the correct option will be 1 fertilization, 2 is cleavage, uh, 3 is also cleavage where it forms the blastula, and 4 will be gastrulation. So because after 1 and 2, since none of the second options have cleavage except option 2, your option 2 will automatically be correct. However, the option 3 and option 4 will also match with your speculation. So I hope everyone's okay with it. So I'll go on to the next question. So sixth question is, you wish to clone a laboratory rat. For that you have taken the female rat egg, denucleated, and now going to put a donor nucleus into it. So which type of cell? So I mean, which type of cell would be best as a donor to take the nucleus from, and why? So this is also straight away from the videos. So we have to take, so if we are going to denucleate a cell and then put in a fresh donor nucleus, then that a cell should be a early embryonic cell because it will be like totipotent. So answer is option 2, which is early embryonic stem cells, because they can achieve the totipotency easily. So moving on to question number 7. The sugar glider, that is Petaurus breviceps, and the flying squirrel, Pteromini, are two remarkable gliding mammals. Despite their distinct evolutionary lineages and different geographic locations, they exhibit strikingly similar adaptations for gliding through the air. This intriguing phenomenon is an example of what kind of evolution. So yeah, already we saw in the weekly recap as well as in the lectures that if two animals having different evolutionary lineages and they have a common function, they're converging towards a common function, so it's called convergent evolution. So I'll go to the next question. So in a population of 1000 individuals, a particular gene has two alleles, capital A and small a. The allele frequency for A is 0.6 and for small a it is 0.4. According to the Hardy-Weinberg equilibrium, what will be the expected number of heterozygous individuals in this population? So they are asking a population of 1000 individuals. I'll repeat the question. And in a population of 1000 individuals, a particular gene has two alleles, capital A and small a. The allele frequency for A is 0.6 and for small a it is zero point 0.4. According to the Hardy-Weinberg equilibrium, what will be the expected number of heterozygous individuals, that is square A, like capital A small a, in this population? So I'll kind of give like 5-10 minutes to solve this question. Like it is a pretty easy one. Like you just have to take in Hardy-Weinberg equilibrium into calculation to solve this problem. Sorry, sorry, the audio mixer actually didn't. I got message that the audio mixer actually didn't work. So I'll re-record this app and upload it. Join it with the version and upload it. So the question is, I'll read the question once again. So in a population of 1,000 individuals, so particular gene has two alleles, capital A and small a, and the allele frequency for A is zero point 0.6 and for small a it is zero point 0.4. And according to the Hardy-Weinberg equilibrium, what will be the expected number of heterozygous individuals? So I'll just take the pen. So we know that by Hardy-Weinberg equilibrium, so your p square plus 2pq plus q square equals 1, where your p is your dominant allele frequency, q is your heterozygous recessive allele frequency, and 2pq is your uh, heterozygous dominant uh, heterozygous dominant individuals number. So here they are given that p is 0 0.6, small a is to q is 0 0.4. So you just have to calculate. So and they're asking expected number of heterozygous individuals. So it will be 2pq, which is 2 into 0 0.6 into 0 0.4. So the frequency will be 0.48, which is 2 into 0 0.6 into 0 0.4. And they're asking this out of 1,000 individuals. So you just have to multiply 0.48 into 1,000, which will give you 480. So I hope it's clear. So next question. In a hypothetical population of endangered turtles, a few individuals find themselves abandoned on an island. Due to the scarce resources and little genetic contact with populations of mainland turtles, their population size has remained modest over numerous generations. As a result, the island population develops distinct genetic traits. Which of the following sums up the idea of genetic drift in this population of isolated turtles the best? So this is also famously theory of evolution from Darwin's theory of evolution. So the answer is genetic drift is the mechanism behind the emergence of unique genetic characteristics in the alien population due to random fluctuations in allele frequencies. So the correct option is option 2. Once again, this is a very theoretical question. So moving on to the next question. Population of wildflowers undergoes evolution over time. The blossoms are initially colored in a variety of hues, ranging from purple to white. Purple flowers have a higher rate of pollination since they are the primary attraction for pollinators. Over a number of generations, the population experiences a increase in the frequency of purple flowers, while the frequency of white blossoms decrease. Which of the following best sums up the process of evolution taking place in this group of wildflowers? So this is like natural selection. So due to the visibility of purple color, the pollination is favored in purple color. So the number of purple color increases. So this is directional selection, where a specific trait is favored, leading to a shift in the population characteristics, where the trait in question in this case is purple color. So that's all the questions are there. Problem questions are there for this week. So I'll leave the room for a 10 minutes doubt session, uh, or a 5 minutes doubt sessions, and from 4:15 we will start the doubt session. Okay, I'll just go on to discuss the answers for week 4 of your set of questions. So, and even in this week 4, we had a completely uh, theoretical set of questions. So we'll just keep going over them. So this week 4 was based on Mendel's genetics and not some cases non-Mendelian genetics. So question number 1: two plants of same species having two different seed colors were cross-pollinated. The resultant project once again. Yeah, okay. So it's So yeah. So two plants of same species having two different seed colors were cross-pollinated. The resultant progeny gave a new color of seeds and none of the previous generation seed colors appeared. What could be the reason behind such result? So this is, yeah, complete case of like incomplete dominance. So the answer will be option 1, two incomplete dominance. So this is a case of non-Mendelian genetics where none of the so this is the case of non-Mendelian genetics where uh, none of the alleles are like completely dominant over the other. One of the alleles will be incompletely dominant, so it will give us a mix of both the dominant allele and the recessive allele. So the answer is incomplete dominance. Question number 2. Rahul is a second year BSc student in the genetics course. He wanted to cross two true breeding flowers, pink, capital P and capital B denoted, and white, denoted by small p. If he crosses these two plants, then what would he expect to see in the F1 and F2 generations? So this is a classical case of Mendelian genetics. So according to law of segregation, all your F1, so the pink is truly dominant over the white. So your if you cross the two plants in the F1 generation, your all your flowers will be like pink in color, and in the F2 generation, three flowers will be pink and one will be white. So the correct option is F1, all flowers are pink in color. F2, 3 is to 1, which is pinkest to white. So this question is also a classic case of Mendelian genetics, and you solve this using your law of segregation. Question number 3. To understand the concept of heredity, different hypotheses were given. Which of the following hypothesis is true about blending hypothesis? So this is also a very theoretical question. So the blending hypothesis like proposes that the offspring's physical characteristics of hybrid of the parents just like mixing of colors. So just a case of genetics that blending hypothesis, blending inheritance says that the offspring's physical characteristics are a mix of the parents' physical characteristics, which is not true. Uh, in all the cases, it crosses over inherits characteristics, which is uh, not true to an extent because in some cases they can recombine and form a completely different characteristic as well. Question 4. If a person with type O blood group marries a woman of type AB blood group, what are the possible blood group types could their children have? So this question is also pretty simple. So person is having type O, so that he'll have IO, and this person marries a woman of type AB blood group. So the woman will have IA and IB. So if these two people cross over, so it will result in the formation of either IAIO or IBIO. So the resultant kid will either have type A, type B blood. So that it's not possible that offspring will have either of type O or type AB blood group. The kid will either have type A or the kid will either have type B. So next question. You came across a maize field and found maize plants with two different seeds, one with white seeds denoted by W and one with yellow seeds small y. You performed a cross pollination between the two plants. The resulting F1 generation had both type of plants in equal proportion. That is 50% plants with white seeds and 50% with yellow seeds. What would be the genotype of plant W for this trait? So noting that dominant trait of white is capital W, and recessive trait is yellow is small w. So here I think there is a miscommunication between the white here and white here and the yellow here and the yellow here. So we should consider that white is WW, capital W, and yellow is small w's. So they are saying that once a certain kind of uh, white and yellow were crossed together, the resulting F1 generation had 50% white and 50% yellow. So this will happen only when the white seeds are like heterozygously dominant. Only then it will produce like two whites and two yellows, like equal whites and equal yellows, which is like a case of your test cross. So your answer will be capital W, small w. I'll once again say the reason behind this. So this question actually is a case of your this question is actually your case of your test cross where your heterozygous dominant plant is crossed with a homozygous recessive allele plant to give 50% dominant plants and 50% recessive plants. So your the question is asking that what would be the genotype of plant white for this trait, which will be like heterozygous dominant, which is capital W and small w. So I'll move on to the next question. So Mithilesh has one black colored guinea pig. He came to know that the trait black is dominant over the white colored trait, which is denoted by small w. But he became curious to know whether the genotype is homozygous or heterozygous. What should we do to determine the genotype of this guinea pig? So that's the thing. So in this case, he should perform a test. As usual, we know that we should perform a test cross. Yeah. So the answer will be to perform test cross. So test cross will tell you whether your dominant allele is homozygous dominant or heterozygous dominant. So if it is homozygous dominant, all of your when you perform a test cross with a homozygous recessive allele, and if your animal in question is homozygous dominant, then all your animals will yield heterozygous dominant alleles. Whereas on the other case, if your animal is heterozygous dominant and if you cross with your homozygous recessive allele, 50% will be your heterozygous dominant animals and 50% will be your homozygous recessive animals. So to know his genotype of his animal, you should perform a test cross. So moving on to the next question. A man with B positive blood group has a child with O positive blood group. Can you determine the possible genotypes of the parents? And I think this we already saw in the previous lectures as well. So the answer will be father is IBIO and mother is IOIO. So in all the other cases, the in the this is option one, and in all the other cases, the resulting child will have B positive blood group. And only in this case, when the IO allele of the father crosses with the IO allele of the mother, the child will also have your O positive blood group. So next question. Ramakant has a hobby of growing flowers in his garden. In the winter season, he planted dahlia plants. The flower position of dahlia plant is determined by axial and terminal alleles. Axial alleles are dominant to terminal ones. Can you state all the possible F1 and F2 genotypes and phenotypes from a cross involving parents that are homozygous for each other? So I'll read the question once again. Ramakant has a hobby of growing flowers in his garden. In the winter season, he planted dahlia plants. The flower position of dahlia plants is determined by axial and terminal alleles. Axial alleles are dominant to terminal ones. So axial is dominant to terminal. Can you state all the possible F1 and F2 genotypes and phenotypes from a cross involving parent that are homozygous for each other? So this is also a classic Mendelian cross. So your F1 will have all axial. Since axial is dominant over terminal, your F1 will all have axial, and F2 will have axial and terminal. Like one second. Yeah, sorry. Yeah. So your F1 will have all axial, which are it's heterozygous dominant, and your F2 will have 1:2:1 ratio of uh, homozygous axial, heterozygous axial, and homozygous terminal. So your answer will be the last option. F1 is heterozygous all axial, and F2 will be 1:2:1 of axial, uh, homozygous axial, heterozygous axial, and homozygous terminal. So ninth question. So you have performed the dihybrid cross. What is the probability of homozygous individuals for both characters in F2 generation of a dihybrid cross? So this is also straight up theoretical, the answer is 1 by 4. Because your heterozygous will be 1 by 2, and homozygous each will be 1 by 4. So they are not asking uh, both the characters combined, it's just both the characters, so both of them have 1 by 4 each. So Sushila had last question. Sushila had an observation that when a white flower crossbreeds with a black flower, it produces grey colored flowers. Choose the correct statement from the observation. So Sushila is observing white color flower, and crossbreeds it with a black flower. It produces a grey color flower. So grey color flower you couldn't observe in both the parental colors. So this is a case of non-Mendelian genetics. So, and notice that this question is also a MSQ question, multiple select answer question. So you can also choose more than one answer. So we can infer from this statement that it's a case of non-Mendelian genetics, and so if it's a non-Mendelian genetics, it will not follow law of segregation, and it will not follow Mendel's law of independent assortment. So these two are the correct answers. It does not follow Mendel's law of segregation, and it does not follow Mendel's law of independent assortment. I'll once again repeat, since it is a multiple select answer question. So a person crossing a white flower, Sushila is crossing white flowers with the black flowers to produce grey color flowers, which is a mix of both the colors. So it's a case of incomplete dominance, which falls under non-Mendelian genetics. And if it is non-Mendelian genetics, it will automatically not follow the Mendel's laws. So automatically, the correct answer will be that it does not follow Mendel's law of segregation and independent assortment. So that's all the questions I have for this thing. So I'll leave this room for a doubt session uh, till 5 or whenever the session ends. And if not, we'll see at the next session will be at the coming Saturday, which is at 8th March from 3 p.m. to 5 p.m. Okay, so if there are no doubts, I'll see you guys on uh, the next session, just at 8th March, 3 p.m. to 5 p.m.